So I'm introducing the speakers on behalf of DAG today. We are going to start with Claire. Is that correct, Claire? You're going to be the first one? Yes. Okay, so Claire is a permanent researcher at the French CNRS, located at the University of Rennes, like Claudia already mentioned, in Brittany. She started very recently in this position this year after spending the last few years as a postdoc in Quebec in Canada. She's an evolutionary biologist interested in speciation and adaptation. With a background in ecology and studying phenotypes, she turned to genomics and now is particularly studying the structural variation in genomes. Her main research at the moment is about chromosomal inversions in seaweed flies, and she's also launching broader projects about structural variation in insects. So Claire, thank you very much to you and Rui. So I will introduce Rui afterwards, and yeah, we're looking forward to see what you guys have to share with us today.
Thank you, Camila. I guess Rui will just take the. We have a mixed presentation, so maybe you want to introduce Rui now?
Sure.
Maybe that will be easier, that everyone see you before I share screen.
Okay, then we're also going to have Rui in a joint presentation with Claire, who is a researcher at CBio, Researcher Center in Biodiversity and Genetic Resources at the University, or connected to the University of Porto in Portugal. He's an evolutionary biologist, primarily focused on studying evolutionary genomics of adaptation and speciation. And he's in particular interested in understanding the role of chromosomal rearrangements in adaptation and reproductive isolation using Littorina species, a marine gastropod, as a model system. So Rui, thank you very much to you as well. So you're going to present together with Claire, the talk about building knowledge in public databases about structural genetic diversity. So, floor is yours.
Can you see my screen?
Yes.
Yes, great. Okay, well, so thank you. Yeah, thank you very much for the invitation. It's a pleasure to discuss with you some of the idea that emerged when discussing with Rui and also with Rebecca, who is here but stay hidden, about what good use can we make of all the resources that ERGA and other initiatives are building to better know about the evolution of genomes and of structural variation. Does it change? Yes. Okay. So just a quick introduction, I guess, familiar for most of you, but what we call structural variant in the genomes are a variation in the presence, position, and direction of a nucleotide sequence. So very often we study variation in the sequence, like single nucleotide polymorphism, but recently we get to know these structural variants. Very often we consider mostly the bigger one, bigger than 50 base pair, but this threshold is really arbitrary. And I guess in the future, we will just be considering all type of variants. So why do they matter? Why are we interested into that? So there is increasing evidence that these structural variants represent a large fraction of genetic diversity. And this is really the basic level of biodiversity. So that's why we want to be able to better describe and understand genetic diversity. So they are not new. We know them for centuries. We knew that there were inversions, for instance, in many taxa, but mostly we knew big variation, big rearrangement, or gene duplication. And recently, genomics and bioinformatics has progressed so much that we have, this is a quick sketch of all the different methods and how we can connect the different way of getting access to the sequence to detect structural variants. And what the studies are showing now is that they cover 3 to 10 more bases than the SNPs. So they represent a large fraction of variation. And it's so important that in some genomes like mice or mussels, there is like a 25% of genes that can be present or absent between samples. So they're really important to consider. Why would they matter for evolution? Also because their mechanism and evolution are complex. They are multifactorial. So first, as we said, they are longer than SNP. so they are maybe more likely to have a strong effect. They can also directly affect gene expression. Let's say if one gene is present or absent, of course, it will affect its expression. And quite importantly for evolution, they may appear more rapidly or perhaps more slowly than the classic substitution nucleotide that we consider. We think about rates of mutation. They also have indirect effects. And one of the most discussed for inversion, for instance, is that they impact recombination. So they mean that this blocks gene flow, and this can create blocks, non-recombining blocks that contributes to adaptation, that kind of contributes to speciation, and really differentiate some region of the genome different that block gene. But there are also other indirect effects, such as decoupling the gene and the regulation, or affecting epigenetic and chromatin change. So there is a lot of research ongoing at the moment, and I'm sure many of you are involved in that too. So all of this research in the last 10 years have shown a lot of large effects of variants that are important for ecological adaptation, for speciation. These are some examples of inversion polymorphism that underlie ecotypes. Quite the famous one of the paper moth was indeed an indel. And there are also examples of structural variants involved in pre-mating isolation or post-mating isolation, so contributing to speciation. And we know that between species, there are lots of rearrangement and change in the genomes. So this is something we have been trying to summarize a few years ago with Rebecca, Anna, and Maren. Maybe this review has aged a little, but I think our main message is what we are trying to do at the moment is thinking that from evolutionary ecology, we know a few examples with large effects, like one indel that does the melanism. Bioinformatics has progressed so much, we are able to catalog structural variants in almost any species. But in population genomics, we still mostly consider variations such as SNPs. So I guess what many researchers are trying at the moment is link these three approaches to catalog all SV, look at them in terms of population, and link to ecology or phenotypes. So yeah, maybe that's the classic about studying evolution, but the idea is to be able to include as much as we can structural variants in the process. So both for population genomics, also for simulation, because if you have ecotypes based on inversion, maybe it doesn't behave like a single species. It's two different ecotypes, but also for experimental validation. So several questions emerged as we were reviewing this work, and I think many of them are still open. We wonder what are the characteristics that make some structural variants important for adaptation and speciation, and why some are not. What are the neutral and adaptive processes that determine the evolutionary trajectory of structural variants? And very importantly, something we really don't know at the moment is how do the abundance, diversity, distribution of structural variants compare across population or among taxa? Can we have a guess about the evolutionary rate of structural variants? And to be able to answer that, we need to be able to compare species and to compare studies. And right now, it's not so much doable because of the lack of data, of course, but also because we lack similar data, and the best practice are just emerging somehow, because everyone was exploring, of course, how can we do? So it's so recent that now we need to homogenize that. So that's why we are discussing today. And I was wondering how can the initiative like ERGA and others can be important for the study of structural genomic variation. So I've put here just a few examples from my research. This is an example in which we study fish in North America. This was Kaplan fish that were studied by Hugo and Louis in my former lab. And when we did a basic kind of structure analysis, we were seeing 3 groups in the PCA, 2 groups in a standard structure analysis. So they were like, wow, there is maybe 2 or 3 genetic clusters hidden here. What happens? And the genome we had a reference genome, but it was super fragmented. So this signal was shared by many SNPs on many scaffolds. So at one point, we look at a very distant species of fish and try to say, do these scaffolds belong to the same chromosome? And indeed, we found that the signal corresponds to only 2 chromosomes. So this means that the 2 or 3 clusters are driven by only 2 chromosomes. So that could confirm that this is not a neutral population structure, but rather a chromosomal rearrangement. And I think this for me showed me how important it is to have chromosome level genomes. I know you are not the audience to be convinced, and that's what you are trying to do with ERGA, but this is why it matters somehow. And another example on fish again is in Coregonus cripiiformis, the white fish. This is something I've been working with Louis and a team in Sweden of Sig and Christina. And we had several different data. We had short reads, we had long read, and we had 2 genomes. So using this data, I've tried to detect structural variants. And long story short, we detect not the same structural variants. And this plot here shows that there is not so much overlap. So that means if one study uses short reads and another study have long reads, we can't really compare the results. And this is what they observe in human. Like a study with short reads, they were saying, oh, you and me, we have about 4,000 structural variants of difference. And then if they return with long reads, well, there are many, many more structural variants. So it's important to keep in mind that depending on the data we have, we won't detect the same variants. So that's why, like, if in ERGA you are doing, let's say, packed value on all samples, that's really a useful data to be able to come back to structural variants. So this problem of homogeneity I was discussing with Conrad this summer, and we were thinking, oh, but Darwin Tree of Life or ERGA, they are generating public data, high quality genome, chromosome level, and also standardized sequencing. Like everyone is doing long read to do the genomes, if I'm not mistaken. And of course, this does just one reference per species. But then what if we map the PacBio reads back to the genomes? So our idea is that we can detect the heterozygous structural variants and also SNP. And we can do that exactly the same way in all the samples that have been used as reference. So this would give really useful data, like make a database of structural variants. This is a pipeline that can be reused if we produce a reference genome from other species. And we can do a lot of evolutionary analysis. So very briefly, what we are trying to start now is to develop a pipeline and validate it, to do a pilot analysis using 100 insect genomes that are available through the Darwin Tree of Life, and then analyze how this diversity of structural variants vary among species. So basically, if we develop this pipeline, this could be something that is the genome after part. So right now, the reference genome from Darwin Tree of Life, they go through deep variants to characterize SNP and indels. And what we could add is detect structural variants. And this could be a pipeline that generates a standard output and some confidence score about like how likely there is a structural variant here. So this is something I was discussing with Mark, I don't know, maybe here, but that we really would like to do with this data now. And with this 100 insect genome, we can test this concept using the data. I also have our Celopa flies, the good databases on Drosophila, and also with Conrad is looking at Z chromosome, and we could make this kind of pseudo-heterozygous data set to confirm that. So then once we have this heterozygosity of structural variant and SNP, we are interested to ask how this heterozygosity, does it correlate between structural variant and SNP among taxa? You must have heard about the paradox of Leontine, that the bigger a population size, the more diversity we expect. But this does not always follow that. Like, I mean, there is less diversity than would be expected in many species. But sorry, there is music. Okay, thank you. So we wonder if deletion or inversion follow the same pattern as SNP. For instance, regarding inversion, I would expect, but maybe that's a wrong hypothesis, but that the meiosis, if it happens in male and female, can determine if one species can be polymorphic for inversion or not. And also, looking at heterozygosity along the genome, we could expect some chromosome to have a higher rate of structural variant than SNP. So plenty of hypotheses that we would be able to test with this kind of really standardized data. So this is just an idea of a pilot study, but of course the longer term would be to expand to a broader phylogenetic scale and a broader working group. So that's why I'm kind of starting and discussing this idea at ERGA today, because the more genomes that come available, the more species we can involve at both Darwin Tree of Life or ERGA or whatever initiatives. And it can fill databases, we can restrict to one sample per species, but then also add pangenome, population level diversity. And in the long run, it would be very interesting to contrast across the tree of life. For instance, in plants, you can ask how mating system affects structural variants. Does it behave the same as SNP? We don't know. So there are plenty of factors that influence genetic diversity, and some of them are likely to influence differently deletion, inversion versus SNPs. So with this, I think I'm giving the voice to Rui to present you another idea he has about what we could do with genomes. Rui, do you want to share screen?
Thank you.
Or you tell me?
Yeah, I can tell you. Just make you some sign for you to pass. Thank you. Just to give a little bit of context why we are doing this together and how this idea appeared is that me and Claire and Rebecca, we're actually working on a proposal together. I think it was more or less how this appeared, and suddenly I was talking that we needed a database on structural variants to explore questions across many systems as possible. And Claire was also talking about the need for a unified pipeline. And then we started to develop those ideas. And at some point we were in contact with ERGA and Darwin Tree of Life to try to see if this was possible within these big data sets being produced. But just to highlight a bit what Claire already said, I mean, we went from the beginning of genetics, we know that chromosomal rearrangements or structural variants as an important role in evolution. For a long time, this was kind of forgotten as we've been studying mostly allozymes and sequences, but not taking into account genome structure. And more recently, when we started to have build these genome assemblies, mainly the ones with high quality, again, we have the chance to look at this kind of variation in the genome. And as you can see here in the right, the differentiation between two sticklebacks, marine and freshwater forms, with the arrows indicating inversions involving their diversions, besides other regions in the genome. They are not only about inversions. And Claire, could you pass just to the next one? Okay. And then we could think this was okay, but it took many years to get from one to the other. But actually things are changing, as you know, and you are contributing for it, or we as a community, really fast. So this review that Claire mentioned before in 2008, this is just about marine organisms because I was interested on this, and at some point I tried to compile some of the existing data, or organisms that live in contact with some marine environment like the intertidal or coastal organisms. And so in that review in 2018, we had like 3 species that could be considered marine, even 2 maybe, if we consider rainbow trout, not strict marine, but actually anadromous. But in like 3 years, we have a lot more of marine species for which inversions have been detected in different species that then have been suggested for playing a role in speciation. So this is happening really fast. And again, we no longer ignore that this kind of variation exists or throw it away because it's sometimes a bit more difficult to deal with. Can you go to the next, please? Can you get to this? Yeah. So just to give an example, and I'm just showing you some slides from some of the organisms I'm studying. This is a marine snail. It's not be considered a model organism in many senses, but it has been more and more used to learn about adaptation and parallel evolution in the shores. And so this is just an example where we were able to detect possible structural variants, in this case, inversions, because they all occur within the same chromosomes, but without yet a very good genome. But I just want to show that is possible, but I also want to, I'm showing this to highlight how good it would be if we have a much contiguous genome reference as we are aiming here, ERGA. So using some population genomics data that we mapped against the reference genome, again, very fragmented, but we had some information to put the context into linkage groups because we had a genetic map. We were able to, using linkage disequilibrium analysis between many markers across the genome, that some genes or some SNPs were linked by high LD to each other in a network. And so we could just identify the regions in the genome where we had high LD, and some of these regions span like 20, sometimes like half a chromosome. And then we could just simply look at the frequencies of these regions or different arrangements across the environmental gradient. And in 150 meters, you can see that some of these inversions change in frequency as the habitat changes. Please. And so we also look at some phenotypes that are involved in the differentiation between the two ecotypes. And when we look to the left and we did some QTL mapping of this, some of these traits, they map to these regions of low recombination that seem to be inversions. And actually, when we compare the divergence between the ecotypes across their distribution in the European shores, you can see that in the bottom figure that, I mean, the inverted regions are these gray rectangles, and they tend to be, the higher differentiation tend to occur in this region. And in the central graph, you can see that inversions tend to contribute a lot to the shared polymorphism of adaptation of these ecotypes across this distribution. So I've been saying we did this, and we have more information from crosses in the lab that suggests that these species, that these regions are indeed inversions. But we actually, at this point, we cannot, we just cannot yet tell you where are the breakpoints and characterize those regions, because we simply didn't have good enough genome. We tried hard to get it, but it was impossible. So we soon will have one, but and others are being produced by the Darwin Tree of Life soon. But this is just an example on how, first, it's good to have population genomics data to infer this process, but still, and you can detect some structural variants that will be important, but still characterize them very well in where are the breakpoints and other things that I will show next. It's still very difficult without a very good reference genome. Yes, you can pass. Sorry. So within other initiatives that actually, at least me and Claire are involved, we've been discussing a lot in lots of brainstorms about inversions, but structural variants in general, not just inversions. And what do we need to know more in order to understand their evolutionary significance across the tree of life, not just if they are important in one or two species, just we showed, but can we try to make stronger predictions, like are, for instance, do we expect that larger rearrangements play a stronger role in isolation or not? Understand better the frequencies. And this is important because sometimes we know a lot, or I think we know a lot about how these processes occur on micro scale, but we don't know then if this shapes the biodiversity at the macro scale. So in order to understand this and to understand how often polymorphic inversions will then play a role in species divergence, or even if that is related with why some plates just radiated much more into many species than others, it's important to start to gather this information. Some have already been published, like even from cytogenetic studies, but you guys are now producing a lot more of this, and we'll soon start producing this at large scale, or already started by the Tree of Life, and soon there will be a pilot and then other projects. And I mean, the idea here would be that I think it would be important to start thinking how to store this data in a way that researchers can use to explore questions about structural variants and their role in evolution, or any other questions that other people may be interested that involve structural variation, like pangenomes descriptions, and so this is just a cartoon. I mean, I didn't go yet into detail on thinking on how such database could be. It's just one example about inversions in humans, but basically I just added ERGA here as an example that we can use already existing data about size of inversions, their location into chromosomes, if their size relative to genome size, and try to get all those sorts of correlations and expect them and understand their role in biodiversity, and also include the new data that is being produced, so that we can not only store that publicly, but that everyone can access to explore questions that go across taxa, and that is pretty much it. I think we have a last slide, but this was just to say, okay, we were invited to present this initial thoughts, let's say, to develop these ideas in more detail later, but to the ERGA community. And if ERGA thinks is something that can be something that ERGA wants to participate or involve people, I think if then you are interested, you can contact us. You can just start discussing this in a more technical detail and so on. Thank you very much.
Thank you so much.
Thank you.
Thank you both. Yeah, really, really interesting. It's great to see ideas coming up to make use of the genomes in scale, so that we can understand more and faster about the biology that we are unveiling now with these projects and many others. So, any questions? Thanks, Claudio.